Scan to unlock Sovi and ace your tests.
Enzyme rate graph question. If enzyme concentration doubles, which curve should the reaction follow?
Enzyme-catalyzed reaction rate depends on enzyme and substrate concentration, so note the setup. Substrate changes across trials, but the original graph used the same enzyme concentration each time. Figure 1, therefore, shows how product changes after 5 minutes as substrate increases. And that pattern levels off because enzymes become saturated when substrate is high and active sites are occupied. Doubling enzyme concentration doubles reaction rate at each substrate concentration, so the new curve should lie above the original while keeping the same saturation shape. Well done! Option B, scan now. Trap, rushing past the given graph. Rule, trust the setup, then check each step before choosing.